Welcome you back. This will be the last session. And in this session, we are focusing on reducing emissions and carbon intensity. So we have the three speakers in the session, and that will be followed by Jay, very popular farmers takeaway from the day session. And the first talk in this session is from Professor Jonathan Lynch from Penn State University. Jonathan is a distinguished professor at Penn State by Penn State University. Jonathan is very well known internationally for his work on the root phenotyping, root biology, as well as the root interfacing with climate change, abiotic stress, and nutrient use efficiency. We are very fortunate to have him as a speaker here. And we'll start with the Jonathan presentation as a live stream. Jonathan, please take over.
Thank you, Raju. Can you hear me okay? Can you see my screen okay?
Yes.
Raju, can you hear me?
We can hear you.
Okay. Good afternoon, everyone.
Sorry about that. Yeah, please go ahead.
Sorry. Yeah. Good afternoon, everyone. I'll be talking today about how we can get deeper roots, which are important for capture of water, nitrogen, and carbon. And so water, of course, water limitation is the greatest limitation to global agriculture. And that is projected to significantly worsen in years ahead because of global climate change. Nitrogen is perhaps the second biggest challenge for global agriculture. Here is a map showing in green the areas in which we apply so much nitrogen that we overload the environment, causing pollution, and the areas in orange and yellow where insufficient nitrogen is applied, and so the crops are suffering from deficiency. In terms of carbon capture, different modeling studies, such as this one from the United States Department of Energy, tell us that deeper rooted crops could have a very significant effect on carbon capture. You can see these are scenarios based on US CO2 emissions. So if farmers adopted deeper rooted crops just in the US, we could substantially offset total US carbon emissions. And so it's expected that there will be carbon credits available for deeper rooted crops. I want to mention first the story about common bean, most important food legume on earth, an important source of nutrients in Africa and Latin America, as an example of successful root breeding. We developed this idiotype called topsoil foraging, which improves phosphorus capture. Phosphorus is an important constraint in these areas. And so on the left, we have a standard phenotype. On the right, we have the topsoil foraging phenotype that's consisting of distinct traits that combine to improve phosphorus capture while also permitting water capture. And new lines based on breeding for this phenotype have been released in Africa and Latin America that have substantially better yield in these tropical soils. As an example, in Mozambique, 6 new varieties were just recently released. Compared with the best existing lines, these new lines have twice the yield, 3.5 times greater household income. They cut soil erosion in half because the plants are larger. They have twice the nitrogen fixation. They benefit women, children, and men in a region of 70 million people. We also did this with soybeans in China using the topsoil foraging idiotype in South China. You can see here on the left, standard soybean phenotype, on the right, a topsoil foraging line that we developed through phenotypic selection. And you can see on the right, much better performance of these topsoil foraging lines. In this case, a region served by 600 million people. 15 new phosphorus efficient lines were released in China, and 3 of them were released in Mozambique. So it is possible to breed crops with better root systems. Now I'm going to be switching gears to talk about maize, which is where we have most of our recent data about water and nitrogen, which is your interest. We have discovered a number of root phenotypes in maize which substantially improve yield under drought, nitrogen stress, or phosphorus stress. I'm not going to go into, we don't have time to go into all these today, but I just want to point out the magnitude of these benefits are quite significant. These are expressed as a percentage gain. So a 100% gain would be doubling yield. And you can see a number of these traits can double or more yields under drought, and several of them have significant benefits under nitrogen stress as well. When we're talking about nitrogen, of course, in a leaching environment, it's moving in the soil. Nitrate is mobile, moves with rainfall down into the soil, which is why roots have to get deeper to catch this before it is lost to groundwater or to the atmosphere. Our idiotype for nitrogen and water capture is called steep, cheap, and deep. It's a combination of physiological, architectural, and anatomical traits. I'm just going to give you a few examples because we have limited time today. So for an architect, so here we can see these two phenotypes. This is top, steep, cheap, and deep, and this is a standard maize root phenotype. So as an example of an architectural trait, I'll talk about fewer nodal roots. So if you excavate maize roots in the field, you see large variation for the number of nodal roots. This is a study where we looked at 5,000 different maize lines in the field, and we saw variation from 5 to 35 nodal roots per plant. Now here we have a computer simulation of these. On the left, you have a line with fewer nodal roots, on the right with more nodal roots. We use this to guide us to the right types of field experimentation, which is obviously slow and expensive. And so here you can see the differences in the phenotypes that we observed in the field. Now the model told us that having too many nodal roots, you can see here 46 nodal roots, actually retarded the depth of the root system, that a root system with 6 nodal roots would actually be deeper because each nodal root would have less competition with its neighbors for internal and external resources. So perhaps contrary to what you might think, fewer roots was better in terms of getting deep resources. This was a computer modeling exercise, but we confirmed it in the field. Here we see drought and a rainout shelter here in Pennsylvania, natural variation in crown root number in maize lines, and yield under drought. And we see that we have, with fewer crown roots, we have deeper rooting, better water capture, and 50% greater yield. This is under low nitrogen in Africa. You can see here in South Africa, this low nitrogen field had chlorosis, as you expect, high nitrogen. Once again, natural variation in crown root number. The lines with fewer crown roots had deeper rooting, better capture of nitrogen, 180% more yield. So that's an example of one of the architectural traits we've looked at. As an example of an anatomical trait, I want to talk about reduced root respiration. So this is not a computer simulation. This is a maize root excavated in the field and ablated with laser ablation tomography, which I'll talk about in a second. And so these yellow-green areas are air spaces that are converting living tissue into air, which should make the root cheaper metabolically. So here's natural genetic variation in maize for this phenotype, Arenchyma, these air spaces. And when we compare these in the field, once again, in Pennsylvania, perhaps like in most parts of Canada, it's hard to get natural drought conditions. So we use rainout shelters here in Pennsylvania to generate drought in the field. And we have lines with less RCA, lines with more RCA, root cortical Arenchyma. These lines are related to each other. They're all descend from the same two parents. They're recombinant inbred lines. And just as we had predicted, the lines with more Arenchyma had cheaper roots, deeper roots, and in this case, 800% more yield under drought. You can see very severe effect of water stress on these low Arenchyma lines and pretty good performance in these high Arenchyma lines. So we validated this study in the field in Africa using the farmers' conditions and farmer genotypes. And so, in fact, the farmers were helping us phenotype, as you can see here. And on farm in Malawi, in dozens of farmer fields, we found that the high Arenchyma lines under natural drought conditions had between 80 and 150% more yield under drought. This also helps under low nitrogen. Here again is our low nitrogen site in South Africa. Natural variation in Arenchyma was associated with yield under low nitrogen. In this case, higher Arenchyma gave us deeper rooting and 60% more yield. So I'm talking here about Arenchyma, which converts living tissue to airspace, which you may not be able to read these numbers, but the first bar is respiration. So the high Arenchyma line has half the respiration, has twice the root depth, and has twice the yield under drought. Now, there are other traits that can give us these kinds of effects. We've looked at cortical cell file number, which is just the number of cortical cells in the root. There's natural variation for that. And when we have fewer cortical cells, We see half the respiration, almost twice the depth, and over twice the yield under drought. Similarly, with cell size, larger cells are cheaper because they're mostly vacuole. There's natural variation for this. And once again, larger cells give us half the respiration, significantly deeper roots, and over twice the yield under drought. And there are other traits we've discovered that we don't have time to get into today, and there might be yet others in canola. But the point is that you've probably heard about water use efficiency in terms of the amount of water used by leaves to gain carbon in photosynthesis, but there's another way to look at this, which is carbon use efficiency. So this is water use efficiency. Leaves are losing water, gaining carbon. But here we're looking at the root side of the equation where we're losing carbon to gain water or nitrogen, and that can be manipulated through these phenotypes. Now, a common concern for folks who are trying to use root traits for breeding is how are we going to phenotype these things in the field? And we have developed techniques to do that. I'll be talking about next. Excuse me. In the field for our root architecture, we've developed something called shovelomics. It's basically digging up roots by hand, rinsing them, and then taking a photograph of the root crown, which can be analyzed by software to generate all kinds of information about that root crown. My colleague Alex Bush has developed software that will do this. It's free software called DIRT, Digital Imaging of Root Traits. You take a photograph of your roots, upload them to his site, and you get back all kinds of metrics about the root phenotype. As an example, this is maize in the field, and you can see variation here for the growth angle of crown roots. Before we talked about the number of crown roots, here we're looking at the growth angle. So they go from shallow to deep, and there's lots of variation for that. In 5,000 maize lines, they go from 20 degrees from horizontal to nearly vertical. So there's large genetic variation for this that is related to nitrogen and water capture. And as you can see, just to make a long story short, to show that this phenotyping platform does work, using shovelomics for a fairly noisy root phenotype in a fairly noisy field environment, clay soil with stones, we were able to identify a gene that controls root angle in maize. Now, how about anatomy? Traditionally, you'd have a lot of work with razor blades and a lab and a microscope. It's very laborious, but we developed something called anatomics. This is a method invented here at Penn State and called laser ablation tomography, which blasts tissue with a laser. This is on the lower right is a real-time image of a maize root being ablated by the laser. The root is vaporized, but then we have all the internal information. This can be used for any tissue or any material, but it works great for roots. And you can see here all the information here about the internal structure of the root. We developed software to automatically process these images. So here you can see maize root cross-section, and here's xylem in yellow, and here's a parenchyma in red, and there's cell size and cell file number and other things can be seen in this image. And this works for dicots as well as monocots. And just once again, to make a long story short, in the interest of time, we did several years of field work with lines with more or less parenchyma, phenotyped them in this platform, and we're able to identify this gene, BHLH121, which regulates root cortical parenchyma formation in maize. This is a very noisy field environment, rural South Africa. And it's a very noisy root phenotype because many different environmental factors will change parenchyma expression. So this is kind of a worst case scenario. If we can phenotype parenchyma in rural South Africa, you can almost phenotype anything using this method. I also want to mention leader, which is specifically geared towards phenotyping deep roots, which once again is a very difficult phenotype to measure in the field because some of the roots may go down 2 meters or more. How are you going to excavate those in a cost-effective, high-throughput fashion? And if you do, you've destroyed your plant and you've destroyed your field. So leader stands for leaf element accumulation from deep roots, and it's based upon this device right here, although you don't need this device. This is a handheld X-ray fluorescence spectrometer, and Molly Hanlon, the lead on this project, is holding it here on a rice leaf, and it emits X-rays into a leaf and looks at the fluorescence of those leaves, X-ray fluorescence, and basically you can look at the entire metal content of a leaf or of anything else non-destructively. And so the idea is you go into a field, you have, in this case, maize, it could be any plant, it could be trees, it could be anything. You have deep roots, deep-rooted line and a shallow-rooted line. In any soil on Earth, because of soil processes, soil morphogenesis, there are going to be elements that are more available to the plant in shallow horizons, and there are going to be elements that are more available in deep horizons. And these are not just nutrients, these could be trace elements. And so by using this in the field, this handheld ray gun, you can zap the leaf and you'll see that in this case, both plants have blue because both plants have shallow roots, but only one plant has red because only one plant has deep roots. That's it. The method is more sophisticated than that, but that's basically the concept. You can also, if you're a company or a breeder, you can put a tracer in your field. We've been using strontium at a certain depth, and then this device will detect the strontium. So the idea is this is a non-destructive way to measure root depth quickly in the field. And we've looked at this in maize germplasm that contrasts in rooting depth across a range of field environments in the United States, many different kinds of soils. And we see a very good relationship between the D95 is an estimate of root depth. It's the depth at which 95% of the roots attain. And you can see a very good correlation, R-squared 0.76, between what Leader tells us is the depth of the roots and what we actually measure the depth of the roots to be via very laborious soil coring. And so we're very easily able to discriminate deep and shallow rooted lines using this technique. So in summary, natural genetic variation for root phenotypes presents excellent opportunities to develop crops tolerant to water and nutrient stress. Subsoil foraging enhances nitrogen and water acquisition in leaching environments. Several anatomical and architectural themes affect the metabolic cost of soil exploration, which is an important component of soil foraging efficiency. The steep, cheap, and deep idiotype is promising. Laboratory and field phenotyping platforms exist. I talked about shovelomics, anatomics, and Leader today. Root focus breeding for drought and low P has been successful in common bean and soybean. We can develop canola cultivars with deeper rooting, meaning better drought tolerance, better carbon capture, reduced nitrogen fertilizer, meaning less cost, less pollution, less energy input, and benefits for producers, consumers, and the environment. Thank you very much.
Excellent talk. And your work can be captured from one quote from your talk. You can phenotype anything. I think that applies across your technology that you developed. It can be applied to any crop species, rooting, as well as maybe other parts of the plants. Thank you very much. Yeah, can you take some questions now? Yeah, we have some questions on the line.
Online. So I should answer the chat questions. Is that what I should do?
No, I think Mark is coordinating that.
Parenchyma are common in plants that are adapted to semi-aquatic. Change the battery. Okay, let's try this one. That sounds better. Okay, thanks for the very interesting talk. So the first question was, parenchyma are common in plants that are adapted to semi-aquatic or waterlogged environments. Have you tested your high parenchyma varieties in high moisture?
We have not done that work because that's very well characterized. There are hundreds of papers about this in the literature. And our environments, and the environments we're focused on in Africa, Latin America, and the US, flooding isn't so much of a problem. But parenchyma, it's well known parenchyma helps with flooding by oxygenating root tissue. We've been looking at it as a way to reduce root cost, to get deeper roots, better soil exploration, but it should have that additional benefit of flooding tolerance as well.
Okay, thanks. Any more from the floor? We have one more quick question online. There's interest in the DIRT website, whether it's freely available to use.
It is, and this was colleague Alex Bush, and we had a project together in which he developed that tool, and that's free. Anybody can use it. So it's just if you Google dirt, I'm sure you'll find it. Well, there's probably a lot of things that will come up if you Google dirt. Google dirt
Maybe with-
and root phenotyping or Alex Bush, and you'll see the software.
Yeah. Thanks again, Jonathan. Please join me now for something.
Thank you all.
So the next speaker is Rachelle McCannell. She's a student in University of Saskatchewan. She's going to present her work on evaluation of canola nitrogen use efficiency, determining proportions of soil and fertilizer-derived nitrogen. Please welcome Rachelle.
Thank you for the introduction and the invitation to be here today. I'm looking forward to sharing some results from my master's research with you all. As we know, nitrogen use efficiency was a highly regarded topic yesterday in the conference, and a highly regarded topic and an important topic as we move forward in addressing some climate change mitigation practices and towards a sustainable future in agriculture. So I know nitrogen use efficiency has been talked about quite a bit, but simply put, we're just looking at the amount of yield that we are seeing over the amount of unit N applied. So basically, what are we getting in terms of yield or biomass over the amount of input that we're putting into the system? And what we've found and what research has strongly indicated is that canola, much like many field crops across the prairies and further wide, are only utilizing and assimilating about 50% of the fertilizer nitrogen that we are applying. And canola is a relatively high N feeder crop, so we're applying relatively high rates of nitrogen that are able to sustain target yields alone in addition to soil nitrogen, but really only 50% of that is going towards the plant and yield, and even a smaller portion of that actually being taken off with the harvested portion of the crop. So where is that other 50% or the other portion of the nitrogen going that's not actually being utilized towards yield? Well, it could be being tied up in the organic matter, as well as the residual nitrogen remaining in the field from or post-harvest of the canola or any crop for that matter, but also in inorganic form, so ammonium and nitrate. And nitrates are more so a concern for us as we want to really reduce the amount of residual nitrate that's in the soil as it poses some environmental impacts in terms of leaching and runoff losses, as well as nitrous oxide losses to the atmosphere. But furthermore, we're also seeing lost inputs by the producer. So nitrogen isn't a cheap input, and we want to really capitalize and be able to utilize nitrogen in the most economic way possible, as well as environmental. So there has been emerging concerns over the use of fertilizer and the nitrous or greenhouse gas emissions associated with its use. And the government of Canada has set forth a goal of reducing nitrogen N2O emissions associated with nitrogen fertilizer to 30% below 2020 levels by the year 2030. And this is really just 6 short growing seasons away and possibly unattainable, but nonetheless, a goal to definitely strive for and maybe surpass in years to come, as well as incentives for adopting best management practices, such as the 4R nutrient stewardship programs. And these all have goals of not only increasing nitrogen use efficiency for the crop, but also improving the farmer's bottom line and utilizing his resources best as possible. So we know that plants, of course, they take nitrogen up from the soil, but is that actually coming from soil that's residual in the or is that actually coming from nitrogen that's already in the soil, or is that coming from nitrogen that we've applied, say at the time of seeding or maybe in the fall? So nitrogen use efficiency, often we look at it in terms of, as I mentioned, output over input, but sometimes what we often forget is about the contributions of soil nitrogen to perhaps the yield we're getting and total nitrogen uptake in the plant. And as we've found, and as research has indicated, as I mentioned, that soil nitrogen is a truly and very important source of nitrogen for the plant, despite applying high rates of nitrogen to the soil. So with this in mind, we wanted to utilize N15 stable isotope tracing. So this allows us to take urea, in our case urea fertilizer, and enrich it with the N15 isotope of nitrogen and apply it to the soil. And throughout the growing season, the canola grows, utilizing available nitrogen, whether that's from the soil or fertilizer, and we're able to trace the fertilizer nitrogen throughout the growing season in the plant and as well as harvest. So at the end of the growing season or any plant samples that we took, we can determine the amount of nitrogen actually being utilized from the fertilizer. So with that, our study objectives were looking at determining the nitrogen use efficiency regarding agronomy and physiological efficiency among a diverse set of canola lines. So this included hybrids, historical open pollinated cultivars, and inbred breeding lines. And what I'll really focus on today in my presentation is looking at the contributions of fertilizer and soil-derived nitrogen sources to uptake among the diverse canola lines using N15 isotope tracing. So our study was over 2 years. We ended up with 3 field sites, 2 in 2022, one of which was at Outlook and irrigated, and the Saskatchewan sites were located on the Low Road Farm at the Agriculture and Agri-Food Canada Low Road site. We had a randomized split block design with variety as the main plot and then N treatment as a subplot. And we also selected sites that had low residual nitrogen. So we're wanting to see the best possible chance of a strong nitrogen response. So our sites varied from 2 to 22 kilograms of nitrogen per hectare in that 0 to 15 centimeter layer. So we looked at a diverse panel of canola lines, and these were lines from the Nested Association Mapping, or NAM, parental panel, and their corresponding hybrids. And their corresponding hybrids were developed by crossing the parental line with a commercial male sterile line. And this is how the hybrids were developed that we looked at. And then we had a fertilized treatment. Our fertilized treatment was 100 kilograms of nitrogen per hectare, and then a control. And we collected plant samples at maturity, and when we collected these plant samples, we partitioned them based on the remaining plant that would be left in the field post-harvest, and then the seed that would be removed from the field. Oop. Oh my gosh. Trying to just go one more. Oh. I think it's just being really slow. Okay, if I can get the figure to show, it's just a figure that demonstrates the diversity of the lines that we were looking at. I'll just leave it there. It shows the diversity of the lines that we were looking at. So they're quite diverse, and some of the lines were just a bit longer season, and that was reflected in the yield results, and as you'll see in the NUE here as well, some of the poorer performing lines were just unable to fully reach their yield potential, I guess, at the time of harvest, such as that NAM 72 line. And This is just looking at basic nitrogen use efficiency metric commonly used today, and very simple, but we're just looking at the amount of seed yield over fertilizer nitrogen. So we overall had relatively low yields, but we do saw variable yields and hence variable NUE according to this metric. So variable between the different lines. However, the variability was slightly inconsistent year to year. So lines perform differently at each site. There was no standout line in terms of NUE according to this metric. Okay, so jumping into the 15N tracing study and results here. So this is looking at the proportion of fertilizer nitrogen that was actually recovered in the harvested seed. So if you look on the y-axis, we're dealing with really, really low proportions, really no more than 20%, and then even lower in 2022 at the Outlook and Saskatchewan sites. And perhaps maybe due to lower yields, but it was consistent year to year, just low recovery of that fertilizer nitrogen in the seed. And Saskatchewan then to 2023 was a quite variable year in terms of lines. There was quite variable results we saw there. And then this is looking at, again, the same metric, but this is looking at nitrogen recovered in the mature plant. So this is what would be remaining in the field post-harvest, and a bit more variable with differences in the lines in 2022 as well at Saskatchewan there. Again, that NAM72 line is just a bit higher as it didn't fully reach its yield potential by the end of the growing season. But again, relatively low, and that's okay when we're looking at the mature plant, as most of this nitrogen at this point should have been relocated to the seed. And now looking at total nitrogen uptake, so these figures represent the cumulative nitrogen uptake in the plant, and in the yellow we're looking at plant nitrogen, and in the blue below we're looking at seed nitrogen, and in the darker shades is soil nitrogen, and in the lighter shades is representing fertilizer nitrogen. And what we really see here is the contributions of soil nitrogen to the total N uptake, and consequently yield as a result. We did see some variability again in 2023. The site was a bit variable than our other 2 years, and some variability in the plant nitrogen derived from the fertilizer at the Saskatchewan 2022 site. But I really like this figure as it demonstrates how much nitrogen was actually being utilized and taken up from the soil, despite the low residual soil nitrogen levels, and applying sufficient amounts of nitrogen fertilizer to provide nitrogen for the plant. And then this is very similar, but just taking the averages of the different lines and looking at total nitrogen uptake in the seed. So very consistent year to year, seeing about 3/4 of that nitrogen year to year being derived from the soil. And again, had to have been considerable soil mineralization going on, perhaps influenced by the canola. But I think the main takeaway from this research I found, and hopefully today that you guys can take away too, is the important contributions of soil nitrogen to plant N uptake. So in conclusion, before I wrap up, I would extend on, thinking about this in terms of nitrogen use efficiency and improving the use of the fertilizer nitrogen we apply. And based on these results, it's not being well utilized, and how can we address that in terms of maybe breeding for increased soil mineralization, and looking maybe further at that or different methods of nitrogen application. Our method of nitrogen application ultimately simulated a broadcast application. So maybe we need to look at different ways of applying and how that maybe would affect our results that we saw today or over the course of our research. So in conclusion, there was limited differences between canola lines in soil and fertilizer nitrogen uptake, and this suggests insignificant relative capabilities. So relative to one another, capabilities of these lines to increase soil nitrogen cycling or utilize fertilizer and soil nitrogen was the primary contributor to total plant uptake despite low residual soil nitrogen levels and sufficient fertilizer nitrogen application. So I'd like to thank the funders of this project and a special thanks to my supervisors, Melissa Arcan, Kate Congreaves, and Sally Vale, as well as the technical staff at the University of Saskatchewan and Ag Canada Farm for all their help in data collection and analyzing. So yeah, thank you for your attention. I'm happy to answer some questions.
Yeah, thanks, Rachel, for your great presentation. Yeah. So you take some questions.
Well, we can start with one from the floor. So thank you for that interesting presentation. Question is, do you think these results may lead to less nitrogen being applied as fertilizer?
You know, they could implicate that, maybe that we should go that route, but I don't know. I don't think I would recommend that in terms of, you know, we apply nitrogen and perhaps we're increasing the soil nitrogen cycling from the addition of nitrogen. I think there's further work to be done in terms of maybe breeding and improving lines in that aspect.
Okay, thank you. Just one quick question. Did you actually, I might have missed it, did you actually look at the soil to see where your labeled nitrogen was, which layer in the soil it is?
Yeah, so after harvest, we soil sampled in the 0 to 15 centimeters, and then, which would be 0 to 6 inches, and then I think down to 18 inches or 60 centimeters. And what we found was essentially below 15 centimeters, there was no labeled nitrogen. We measured N2O gas emissions the following year, but we didn't have any isotope tracing for that part of it. Yeah.
Okay, thank you. There's no further online questions. Any from the floor?
Yeah, thanks for the presentation. I'm just wondering if there's time left in the project to look at the labeled fertilizer, like labeled N, in the upcoming season? Because so little of it was taken up in 2024 or in previous years.
Yeah, and I'm not a part of, or I've done my portion of the project, but we did take additional samples summer 2024 in the same spots that our previous microplots were, and then we have the data, I guess, or the imaging to know exactly where they were, and there might be follow-up studies or research looking at, you know, the fate of that nitrogen.
Yeah, this concludes this session. Please join me thanking all three presenters of this session. So now I invite Jay Whetter to moderate farmer takeaways from the day.
Thanks, Raju. Came to life, right on. Thanks, Roger. Cody, Alan, and Chuck. I got 1, 2, 3 here. You guys. guys can share that one. I'll send it. I heard some comments how much people appreciated this quick 5-minute at the end of the day to hear from the farmers. So thanks, guys, for doing this.
You're welcome.
All right. What was the most interesting thing you heard today, Cody?
Winter canola. The whole idea behind it is intriguing to me. I think we focus too much on how to get canola to handle the heat better, and maybe we should just shift it to a flowering period when we're less likely to see the heat.
I think I had two really interesting things I heard this morning. First was the valorization of canola for bioproducts, just finding new markets for the canola meal as we increase our domestic crush. Also, I really like the opportunities of canola meal in food products. So again, another option for value adding to the meal and the products that we produce.
Yeah, one of the things I picked up on that, Chuck, was the using canola meal for a seed coat or for a coat for the fertilizers. I think that was one of the things.
Like mean,
yeah.
From the straw, the coat fertilizer.
Yeah, exactly. Kind of cool. Alan?
Well, I had them too. I wrote down quite a few, so it was a great day. And just quickly, so yesterday I was kind of negative. Today I am super positive. And like Tegan was chatting about being Dorothy on the yellow brick road this morning. I kind of felt the same throughout this whole, maybe not Dorothy, a little bit more like the Tin Man on the yellow brick road, but basically going through this whole process and looking at it as a producer, I'm sitting here and I'm going, you need a team probably to get to Oz. We've got a hell of a good team on the floor here, and as a canola producer, I'm glad because this makes my world of production a whole lot simpler. But basically, so here's what I did pick up. Sclerotinia resistance, huge. Root architecture, I think, is going to be very necessary to climate resilience or climate adaptation. And 50 inches of rain in one growing season. Where I come from, 50 inches is 10 years.
You have a heart, Alan, so the Tin Man analogy isn't quite right, but we'll get there.
I think it's very close.
All right, Chuck, you can start with this one. I'll let you go any way you want with this, but I was going to ask you, from what you heard today, anything you'd like to learn more about? Maybe there's a lot of things. Research more, or something I should write about?
Well, if Alan feels like the Tin Man, I probably feel more like the Scarecrow because I really need a brain to understand what was talked about today. But I think there's two things that I really recognized as very interesting, and I think I would really like to see a lot more work being done to it. And the first one is the biopesticides dealing with the sclerotinia, and also the work that was being done with the disease resistance genetically, which I think is also very important as a way to reduce costs for producers. And the other one, like Alan, the deeper roots, the structure, the root mass and all that, I think that's a very good way to go in the future here when we're dealing with, sequestering carbon, improving uptake of nutrients, water management. All those things are very important in producing a crop.
Yeah, I liked seeing the diagrams of the big roots. So we got roots and we got sclerotinia. Alan, we'll come back to you, and Cody will end with you. Alan, what do you got for the things you want to learn more about or research?
Well, I thought you could probably write an article on canola's value in the green economy. So there's a lot of different things, and I think from my perspective, and probably farmers, it's good to know that we've got so many opportunities that do arise from research into canola. So there's your challenge.
Thanks, Alan. Cody.
An economic impact of increased nitrogen use efficiency. I wonder how that would look at the farm gate. Year one, I guess you'd see more uptake would mean less nitrogen fertilizer, but what happens in subsequent years to the N that's in the soil?
Right. Did that second last presentation make you wonder?
It piqued my interest, yeah.
Yeah, I want to talk to the McCannell. What was her first name? Sorry, Rachel. Rachelle? Okay, sorry, Rachelle. I do want to talk to you at some point about your work. Before I let these guys go, does anybody have one last question for the farmers? Anything you really want these three people to answer, or do you want to just find them later? Okay, you got one? Quinn? Yeah.
I threw my hand up and now I got kind of scared. It was just a instinctive thing. What sort of opportunities do you think coming out of this whole week of research opportunities and research with canola, coming from a producer perspective, like on farm, what do you see in the next few years that producers can be doing to improve their canola production, just very generally?
Alan, do you want to start?
Tough question. If we had a weather dial, I think we'd probably have it built right there. Production-wise, well, I think probably there again, we've talked to 4R and that type of stuff. I think a majority of producers are actually practicing that type of system. So get out there, soil test, figure out what you need to put the right source in, right time, right place, all of that. What else can we do? Buy the right type of seed. Basically, I think probably continue doing what we're doing with a view to hopefully adding better genetics that maybe can withstand some of the climate variability that we're facing in this day and age. So probably good luck.
I think really it's just continuing to do what we're doing now, just trying to be more efficient. I really think that greater adoption of the 4R programs, making better use of some of the enhanced efficiency fertilizers, anything we can do to help either make our crops more efficient when we're producing them, or what we can do to reduce emissions to deal with climate change and things like that. I mean, climate change is a really long-term project, but if we can do anything to get that started and slowly reduce the carbon out of the air, whether it's through sequestration or reduce emissions, I think that's something that everybody has to work at.
Cody, last word to you.
Yeah, I don't really have anything to add other than just as farmers, make sure we're doing all we can to give us the best start. An older fella at home said, just if you have a crop wreck, just make sure it's not your fault. So I guess just as long as we do what we can, and if there is a wreck, we get to blame the breeders, which is always easier. So just do what we can, hope for the best.
Right on. Thanks, guys. I got one last thing to say. Just personally, big thank you, while I've got the mic, to Sally and especially to my colleague Taryn. Taryn has been working on this for months, and she stayed up till 2 in the morning the past 3 nights getting everything ready, while I was had been in bed for many hours. So thank you very much, Taryn. Great job.
Okay, just a few things to wrap up here. So again, thanks so much to all of our speakers. We had a huge number of speakers, over 40 plus this extra farmer wrap up at the end of the day, which has been really great. Thanks, Jay. So just thanks to all our speakers for making the trip out and all your prep and having great presentations so that we fueled all that engagement from the attendees. And thanks to all of your participation through all your questions and just discussions on the side to kind of keep the energy up around this. I've mentioned it before, but CCA credits, if you haven't done it yet, make sure you do, either with the QR code, which is up on the screen now, or at the back, or scanning it on your name tag. And then just a few acknowledgements from me as well. So again, thank you to the Canola Week Organizing Committee for all your work over the last year. I'd also like to thank Chris Matcher for your work coordinating the poster session. I think that worked out really well, and it was great to see all the students and other poster presenters here. And to Jay for all your work and creative contributions I wrote down. Much appreciated, and the rest of the Canola Council staff that were pretty involved as well. And of course, to Sally, my co-chair, great job, and thanks for all your efforts. I also want to give a big thank you to Spark. So Caitlin, that you've seen at the back, and Aaron, and then Terry as well. They've done a lot of work on this event, and they've been really great to work with the whole time. So big shout out to them. Thanks so much for that. If you're ever organizing an event and you need some extra help, I highly recommend Spark. I would also like to thank TCU Place and their staff for being a great host for Canola Week over the last few days. And I especially enjoyed the hot apple cider that hopefully some of you got to enjoy as well. And then I would also like to thank our event sponsors. We really couldn't do this event without all that sponsorship, so much appreciated. And with that, I'll close this year's Canola Week event. Wish you all a safe trip home and a great rest of the year. Thank you.